Hello everyone. We will wait for a few more minutes for people to join. I see six people have already joined, so we'll wait for a few more minutes. Okay, I see nine people have joined, which is good enough for us to get started because anyway, in the beginning, I'm going to recap what we did last time. Okay, so where are we now? We were talking about germline mutation and somatic mutation, and we did quite a bit about how do you find germline mutation from doing variant calling. Some of it you might have done in your hands-on exercise in the class. Some of you may have to catch up, but I think all of you may have some idea what a germline mutation, how do you map the reach to the reference genome, and then how do you create a VCF file which has variant information, and how do you find individuals varying in what, whether they have a germline mutation that's known or unknown or whatever. Okay, so we ended up with saying that we can also do variant calling on an entire population, like say 100 insects or 100 human beings from various ethnicity, or 100 plants which grows in different places or which has different kind of traits, like one is tall, one is yield is high. So variant calling comes in every part of our life and in every application, like from plant breeding to cancer diagnostics to cancer treatment um, to genetic engineering, where you want to find regions where there are no mutations in an entire population. So when you're doing that, then you have multiple variant calling files, and that's where the arithmetic comes in. I'm sure in the class you will be doing it. I already told the seniors that they will give you few VCF files to practice. How do you find intersecting variant that are common, variant that's not common. So I'm going to now um, switch to my PowerPoint presentation, and this is where we were last time. Arithmetic of variance. Variance common to two individuals. That will be intersection, correct? You can use multiple tools, as I said. You can use bed tools. Let me pick a pen here. I think we did. Oops. Yeah, I think we did this last time, but it's okay to repeat for people who dozed off. Bed tools, VCF tools, and number of them have an intersect function. What it does, it takes multiple VCF files and intersects it and gives you those that are only common. But then you sometimes want to know variants that are unique to two individuals. Okay, then what you do is to subtract one from the other depending on who you're interested in. Suppose, as I said, between my brother and me, I want to see what is unique to me because I have some medical condition that he doesn't. Then I need to subtract my brother's variant from my variant to know what may be causing my condition. Correct? So, subtraction is also one function that is there within BET tools and VCF tools and BCF tools, whatever. Okay. So I'm dropping these names because now you can search for command line and you can get very smart about how you run it. Okay. And then there is this what I didn't mention last time, variations across population. So when you're taking 100 individuals, I want to know where in the regions of the genome where it's pretty conserved, like there is no variation. That means it's naturally selected to not vary that much. Why would I do that? Why would I want to know about that? Those are for engineering something. Especially these days, you have this technology called CRISPR-Cas, CRISPR-Cas9 technology, where this, if you want the entire population to have a benefit of something that you're going to be inserting, you want to insert in a region that least changes evolutionarily. Okay, that is where this comes in, variation across population. So you may be interested in most varying region also at one point, some application. You also are interested in most conserved regions, which are where you can safely insert a CRISPR cassette with necessary genes in it and make sure that during evolution that will not get kicked out because something got mutated. Because the region that you put it in was very conserved across the population. That's where that comes in. So let's move to the next slide. Okay, so but this is the point you have to remember. When you're doing variation, going to compare hundreds of samples, variants from hundreds of samples, hundreds of individuals, you want to make sure that all of them have been mapped originally to the same reference. Because this is the point sometimes you miss, and you say, okay, here is a reference, I'm going to take it, and someone else mapped 50 samples on some other reference, and you mapped the rest of the 50 samples on some other reference because he didn't know what they mapped onto. You need to really know because otherwise they are not comparable. Okay, that's an important point to know. Especially in human, you have HG19 and HG18 and HG17 and all that, but you have all these different assemblies, right? These are different assemblies, and of course, this is the most popular assembly. So, when you're trying to do individual variation among human, you will have less confusion because majority of us use HG19 because that has been standardized. There's a lot of annotation, which I will tell you how to go about those annotations later in this lecture. But this is the tricky thing when you're doing your own assembly of your reference. Because what happens? Because yesterday, say, Suvarta or Afia gave you a reference, and today someone else improved the reference, and they're giving you the reference. You have to make sure that even though there is a better reference, you cannot start suddenly using the better reference unless you're going to map every sample to the better reference because worse reference genome is better for you because that is where already you have invested heavily and you have mapped, you say 70 samples and you have 10 more coming. You have to use the old reference that you used even though that is not best reference. This is very, very important to remember, especially for students who are doing assembly on one side and mapping on the other side. You can't arbitrarily use a new assembly for mapping. Why would that be the case? Why am I making such a big fuss about it? Because look in the bottom of the slide. You have a reference 1 to 1000. Suppose that's That's the length of a contig in your assembly, and this is the old assembly. Okay, when you did the new assembly or you improved it or something, suppose you found an insertion and there's a 250 base insertion in your new assembly. Okay, and now the same contig is now 1250 bases. No problem. Mapping program will absolutely map it perfectly because it has the same region. Maybe it will map a little bit more in this region as well. In this region as well. Yeah? But what's going to happen is the numbering changes. Coordinate. We always talk about coordinates in SNP calling. Okay? Coordinates are important. Coordinates are always with respect to your chromosome. So you say chromosome number, and coordinates precisely tells everyone which mutation you're talking about. Okay, so, but if you didn't tell them with respect to what assembly, then we're not comparing apples and apples, we're comparing apples and oranges. So, for example, the mutation in the top assembly, suppose you find a mutation, this is chromosome 1, and at position 403, you are finding a mutation. Okay, and then someone else is using the new assembly, and they're finding a mutation at 653. They are the same mutation. They are absolutely the same mutation. But when you're doing the arithmetic, it has no clue. It is looking for 403, and if 403 is not there, it's going to say it is new. It's going to say this is not common between the two. I think you're getting it. Many people might have already known about it, but you're getting it. When you use different assembly, you will not find any common, although you use the same sample. Use one, you may want to do your own exercise on that. Same sample, use one assembly and use the next assembly. There will be no mutation that's common according to arithmetic. That's why I'm saying arithmetic, because arithmetic is a number game. If this is 403, it doesn't know 653 in this assembly is the same as this. That's why I'm hammering on the point that when you're doing hundreds of samples, when you're merging files and when you're doing subtraction, when you're doing intersection, you want to make sure the VCF files were created with the same assembly. This is a very important point, okay? Okay. Now, what did I mean by... Are you able to see this slide clearly? I'm not sure because for me it is too small, but on the top, of course, there is genome, and even on the top here, there are these two exons, right? Two pink exons, and this is the genome. Right in the middle, two spinal things are chromosomes going in the opposite direction, and on the top there's the 3 forward frames, and on the bottom there is 3 reverse frame. I talked about it before, so you know it, I think. Okay. So, and then I have this, if you don't see it, that may be not that good, but I'm just going to see, maybe, because this is I'll tell you where you go to see this. 3.92 point, I forget, someone who knows that can type that in on the chat. This is our JBrowse for ticks. I forget the number. Oh no, I have it right in front. 125, 130. 93, 125, 130. When you go to that, this is the browser I'm showing you right now. So when you go to that, you will see this, okay? Of course, you have to turn on something, turn off something. That's not as clear. So here, where I put the red dot are the SNPs that are common to 90% of the samples. Okay, that means if there are 100 samples sequenced, 90 individuals have these SNPs. Obviously, you can see that here there's nothing. Okay, there's nothing common to 90% of the samples. Then you drop down to 80% of the samples. Sometimes that's good enough. Okay, 80% of the samples, you got one SNP here, one snip here, one snip here, one snip here and here. Yeah? Okay. So the number of snips is going to change, going to increase as you lower the bar. Okay. Now you have 70%. This is 70%. This is 80 and this is 90 in case you're not able to see it there. These are very small. 70%. Then immediately you got 1, 2, 3, same thing, and you got one extra. Yeah? You got one extra here because this is common to 70% of the people. So this kind of display nicely tells you where are the gaps. These are the gap area where there are no SNPs. Okay, where you could put a CRISPR-Cas. And it's also in the intronic region, right? This is intron. So it's safe to put a CRISPR-Cas. So it shouldn't be in an exonic region. For example, you don't want to put CRISPR-Cas right here, although there is a gap, because it's right smack in the middle of the exon. So when you're picking all this, molecular biologists and biologists, they need to pick it smartly so that when they insert something, they are not disrupting the function of this gene, because this gene is a very important gene. Okay? So now you can see why in 70%. Let's see why this showed up in 70% and didn't show up in 80%. Okay, so you can see that this is the variant here. This is not there in this sample. In the second sample, it's not there. It's there in the first sample. It's there in the third sample. It's not there in the first sample. So it's not there in 30% of the samples. That's why you're not seeing it in 80% and 70%. So that's one way to visualize and get regions that are, you could do 1,000 samples, then you get even better results, right? Yeah. So there are merge functions. So for getting these kind of files, okay, you get a, anyway, that's also in bed tools. Bed tools, merge. And you give number of VCF files, it will merge it. And in the VCF file, at the end of every row, it will tell you this mutation is there in this many samples. So you can parse that and get the 90%, 80%, 70%. That's why you need to know a little bit programming to get all this going. Okay. So, for example, I'm just showing 3 individuals, just so you know. I'm pretty sure everybody is familiar with union and intersection, but in case you didn't think about it too deeply, this is the time to learn. Like just 3 samples, sample 1, sample 2, and sample 3. Okay, so when you're doing the bed tool intersect and everything, only 23 mutations are common to all these. That's all the intersections. So if you're only interested in common SNPs, then you got 23 rows in your VCF files. Okay. But if you're interested in the union, this will be the union, all the SNPs in all these 3 files. It is not the sum of all the VCF, all the SNPs, because some of them are common. Common SNPs will shrink into 1 row. Okay, the total number of rows you will have. So although sample 1 has 3 plus 4 plus 23 plus 17, that is how long the VCF files are. This is how many variants in VCF for sample 2. This is how many variants in VCF for sample 3. But the total won't be the total of all the VCF, correct? I know you guys know it. I'm just hammering so you understand my next step. Okay. So 23 is common to that, so there will be only 123. 17 is common to 2, there is only 117, 25, and so on like that, right? So this is the union. Union of all. That means no variance is repeating, but some variants can be in more than 3 samples. You can create this using the merge. Merge program already does the union. It doesn't just, so between concatenating, why I'm telling is that there may be temptation to concatenate the VCF files to get a file of all 100 samples. If I say that merge all the files instead of using bed tools, you can just concatenate it. Concatenation will not work because it will have multiple same variant will repeat multiple times because it's there in multiple samples. If there are 100 samples, it will repeat 100 times if it is common to all 100 samples. That will be crazy. So you need to get union. And union is different from concatenation. So in union, there's only one representative for every variant. Okay, even though it may be there in 100 samples. So when you do that, merge routine in bed tools and other tools just does the union nicely for you. Okay, it's not a simple thing, but it does it very nicely. So you don't have to worry about. But do not concatenate it when I'm saying that let's merge all the files. You just concatenate and merge everything. Don't do that. So after you merge, after you got the union, then how are you going to see which sample is closer to what? How they have diverged because you want to know the whether two samples are identical, two samples are from the same trait, two samples are from different trait. How are you going to get that signature? So you create a huge file, like suppose you had in your union you had this many rows, okay, then this matrix will have that many rows, as many VCF rows as the union file will have, as the merged union of all the VCF files. Each row then will carry the sample information because each variant, this is the variant here, this is the coordinate of the first variant. It comes from the scaffold 1 and the coordinate is 746. Okay, at this position, and here it kind of wraps around. You see the sample information. So you have chromosome, you have position, and you have sample information. Sample 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 samples. It is wrapping around just because I couldn't get the Excel sheet here. So, but this belongs to this sample, this belongs to this sample, this belongs to this sample, and so on, and this belongs to this sample. Okay, so that's how it goes. So now when I have a 1, that means this variant is present in sample number 3, MQ3. It's present. It's absent in MQ2. It's absent in MQ1. Okay, so that's how you fill it. So you now take a VCF. I'm going to call it merged VCF and create this kind of matrix. Once you have this matrix, you can use HCCLUST and everything in R. I think R has these tools, and the senior students will tell you what are the steps to get this matrix, and then go into R to create. Now when you have this matrix has all the information of evolutionary divergence of all these samples. So if you have 100 samples, you can imagine there will be 100 columns. And if you had, say, 10,000 SNPs from the union, then you have 10,000 rows. So this will be usually a huge matrix. Okay, huge matrix, but you don't have to worry about it. There are tools to handle it. Okay, but you do have to create this file and make sure you created it right using the right kind of tool. So once you have this matrix, then you can say that, yeah, I'm going to create a tree. I'm going to see which samples goes together, which samples diverges together. In this particular case, you can see that all these samples are in one clade, all these samples are in one clade, all these samples are in one clade. Some of them get mixed up. This is kind of bad. Maybe the samples got mixed up, or maybe the signature is not decent enough. So you got clade by clade, you know which mosquito, which insects or which plants are going together. Okay, so that's one way to visualize what's happening. Okay, and even to say that your variant calling is working fine. Okay, and now you can get a signature out of this. I said I get a signature that is unique to this population of plant, insect, or human being, ethnicity, whatever it is. You can say that all these people probably come from the same place, all these plants have similar trait, Now I'm going to get a signature out to know that what the trait may be resulting from. Yeah. So here we have used both the heterozygous mutation and homozygous. I hammered on this many times what are heterozygous and what are homozygous. So homozygous alternate alleles are what is used here, and heterozygous is one arm has a reference and one arm has a different allele. So both has been used here. So as I hammered in my previous lecture, the homozygous alternate alleles really carry the information on natural selection. Natural selection selects for those mutations that makes the plant survive in a different neighborhood, in a different environment, and hence propagates that. When it propagates the same mutation that has a benefit to the plant or benefit to the insect, then it will become homozygous in the resulting population. So the homozygous mutations carry the differential homozygous, let me see, population 1 to population 2. The differential homozygous mutation in these two populations really tells you what got naturally selected to cope up with this different environment or different traits. So, in the next slide, I'm going to show you what happens when you do the homozygous. This tree is slightly different. I think these trees can also be generated in a different way. Here, in this tree, there's a big mush in the middle. You can't tell which one is going where. Although you can tell the yellow is nicely diverging, green is nicely diverging, but many places you don't know what's going on. This is not giving you a clear picture of the distance between the two clades. Yeah? So one of the ways you can do, there are many ways you can create tree. This is called rooted tree. This is called rooted tree. And the next slide I'm showing is called rootless tree. Okay. This is no roots. Okay. You are not assuming any you're not assuming any of the you didn't do it right. Okay. You're not assuming that any of the trees are special to you. You're saying that just tell me what's happening between all this. And so nicely now you see that boiled from city 1, boiled from city 4, nicely. And this distance tell you that this is the distance of these two wild ones from the rest of them. Okay. Of course, there is one here, one magenta, just because it had very low coverage. So it was a wrong sequencing error. Okay. Sequencing problem. Okay. This because of sequencing problem. But rest of them nicely going there. Now there is one lab strain. You see, this is not a bad lab strain. Okay, they are all going together from CT2. Okay, there's one cluster right here, very close, and then the others have diverged. This is a good cluster, and still I think one of the greens went here. Whatever happened to that, we don't know. Okay, but rest of them cluster here. Then you see lab CT3 goes right here, all the red nicely, and then there is some mix up here of the red and green, but rest of them goes there. So you see, but what this vertical distance tells you, that is how far apart each clade is from each other. So obviously lab CT3, CT4 and CT1, they are much closer to each other than CT2, wild, CT1 and CT4. That much is clear in this. But we have used here only the homozygous alternate. Not differential, whatever you call it, okay? Alternate allele. So that's what we use. This is one way to look at it. Now you can go ahead and say, okay, I'm going to get a signature for CT4, signature for CT1, which makes this population really distinct, and see whether that has anything to do with the environmental aspect of it. If it's an insect, is there insecticide that has been used in CT1 that's not used in CT4? And what's the history of insecticide in these cities? Those things you can correlate, and just sometimes you just need signature to even differentiate whether some population is coming from that population or not. If there is mixing up happens in the lab, something like that. So anyway, so this is one way to do that. I don't know whether in this summer course you will be able to get this far, but still you know why you're doing the variant calling sometimes and how can you get the population genetics going. I'm already 30 minutes into the thing, and then I'm going to go to the next slide. So these are a little bit different way of looking at it. So these are homozygosity index. So you have a 100 kb length of, so you go as a window from one end of the chromosome to other end of the chromosome, 100 kb, 100 kb, 100 kb, 100 kb, and then see how many homozygous mutations are there. And this will be called homozygosity runs. The square ones, so what do you expect? The square ones are the wild type. Okay, square ones are the wild type and circles are lab. So in this plot we are showing how many homozygous sites are there when you go across a particular chromosome or contig or whatever you have. You see that the wild type has lower homozygosity index, which are all here in this population. Lower numbers, all the square ones are here. One of the square went here for some reason. Okay, but you see the most of the circles are here. Those are all lab strain. Okay, lab strain, of course, some lab strains are very low ones like these ones. Yeah, these are very low, same as wild type. But majority of them are drifting to high level of homozygosity because they have been bred in lab and they're more likely to converge. Because they're lab strain, they breed within the same population, so hence there will be more and more homozygosity that accumulates in the lab strain. So we feel like okay, we are doing good here. At the west, it tells you that the city wild type and the lab strains are behaving as they should be behaving. And then I talked about fixation index. Remember that was in the last lecture. I talked about fixation index. The fixation index. What we found here. I'm going to go back to the slide. Why we did this fixation index is because you see that the lab strain, although they are coming from different strains, okay, they're very close to each other. We didn't know whether that is some artifact or what's going on. So we wanted to use different tools to show that it's not artifact, this is exactly what's going on. So either the lab strains are converging for a lab environment in one way or the other. So this was a very interesting observation we did. So when we did the fixation index, what was fixation index? Whether two, it tells you whether two population has diverged or not, right? Whenever we took a lab, and here you see, whenever we took a lab and wild type, the fixation index was very, very high. When we took a lab and lab, it's low. When we took lab and wild and wild, it was a little bit higher because wild types actually diverge. So this shows you that the fixation index is also consistent with the tree we mapped. Okay, so these are kind of analysis you can do in the population analysis. Okay, so again you have principal component analysis. Principal component analysis again tells you the same story. Okay, one of the wild type is here, one of the wild type is here, but all the labs are kind of mushed together. Okay, so the same thing what the tree is telling us. So we know something is going on in the lab strain. Anyway, so this is not about so much about what we are working on, but just to understand how far you can go to do the population genetic just from variant calling. Okay, simply variant calling, if you know how to do the arithmetic, you know how to do the clustering, you know how to do all that, then you will be able to actually address biology. Okay, so I think you're getting the point of what variant calling can be used, how you can use the variant calling for population genetics. And of course, I haven't gone into the details of any algorithm or anything, but just you know how far you can go and you can learn on your own, correct? Okay, so this is a slide from way back, the third or second or third lecture I did, where now mutations can also, one of the ways you can do, suppose a particular mutation is homozygous, especially homozygous mutation within a population that's unique to that population, and you're interested in knowing why did it pick it up? Why did selection, natural selection pick that one? So you want to look at the impact of that. Is it there in an exon? Is it changing the amino acid? If it's changing the amino acid, does it have any functional role? Is it changing the function of that particular mutation? Those are things you want to ask now because you have a signature. You have a homozygous mutation that's unique to city one or unique to a population, unique to ethnicity. You want to know why did natural selection selected it in the first place. So I think I told you how we can go. I showed you how painfully we went from a mutation to what it does to the amino acid. Yeah, we did that. I'm not going to repeat it. And which codon is it changing? It is going in the opposite. So it is GAT, GAC becomes a GAA, and so aspartic acid becomes a glutamic acid in this case. That comes from this table, codon table. We did this, so I'm not going to hammer on this point. But suppose you have 3,000 homozygous mutation in a population. Are you going to go one by one like this? No, you can't. You can't do this because this is crazy. Because it takes time. Okay, so if you're interested in one mutation, you could do this. Sometimes, when large number of mutation you have, you automate it, but you want to test whether your automation was okay. Then you can go back and cross check. This could be a good validation tool. This can be a good prediction tool when you have one or two mutations. But when you have within a population your homozygous 100 or 200 homozygous mutation, this is not the smart way to go. That is why bioinformaticians need to know the tools. So sometimes what happens, bioinformaticians know the tool, but they don't know why they're doing what they're doing. That's why I'm teaching you how to do it by hand. Molecular biologists will know how to do it by hand, but they wouldn't be able to automate it when they get a large number. So molecular biologists need to work with bioinformatics to get what they want and then go to the lab and validate that. So how do we do automate this, right? Okay. Okay. Here is a small exercise. I don't know whether you'll be able to see from the top here. Okay. So in the automation, what are the issues in the automation? Okay. Um, I know I'm going a little slow, but we do have another lecture tomorrow, so maybe we can continue. One of the most complex thing in SNP annotation is finding the reading frame for each exon. Okay. Let me blow this up a little bit. Are you able to see this? Can anybody type in to see whether you're clearly able to see this part? See this part on the top? Okay. What I want you to do is to, okay, this is an exon 1, the pink one, and this is exon 2. Yeah? Okay. So anybody typing that you can see it, you cannot see it? Can anybody put in the chat? You're able to see it clearly, the three reading frames above and below? Because I'm just going to do a little bit quiz for you, okay? So you know the beauty of what it takes to annotate large number of SNPs, saying that it is not in the intron, it's not in the exon. If it is within exon, it is changing this amino acid to this amino acid. This is not an easy thing. Okay, SNP annotation is a hard one, and there are tools to do it. Okay. So nobody is answering me on the chat. I don't know why. Are you guys able to hear me? I just want to know whether you're able to see the top portion of this slide. Okay. So if you're able to see, just say that we are able to see. Okay, one of you. Afia, can you do that? Can you just say whether you're able to see it? Okay. So you have two things here. Okay, the first thing, the reading frame for the first thing, that, of course, this you know already that this gene is coded in the opposite direction, okay, in this direction. So the reading frame are not the top three, but the reading frames are below the genome. Okay, there's one, two, and three. One, why am I not able to? One, two, and three, right? Three of them. This exon, you can tell can because there is stop codon. I told you if the stop codon within an exon, it cannot be a reading frame, correct? So this exon is in the sixth reading frame. 4, 5, 6. Top 1, 2, 3, it's not because it's not going in the forward direction. It has to be the bottom. It's 4, 5, 6. It's a sixth reading frame because within the exonic region, there are no stop codon, only in the sixth. So it is sixth reading frame. Okay, so it's 1, 2, 3 is what usually used. So it will be like a 0, 1, 2. 0, 1, 2. So it will be the 2 in the reverse direction. Okay. Then you go to this exon. They are coming from the same gene. Okay, in this exon, it cannot be the third. It cannot be the sixth frame because in the sixth frame you have a stop. Okay, in the second there is a stop here. So this exon translates in the fourth reading frame. Because there is a stop codon here in the fourth reading frame, but that comes before the exon start. It is before that. It's right here somewhere. Yeah? So within the exon there is no stop on the fourth reading frame. So do you see that every exon within a gene okay, so we'll do that. You can go ahead and when you go into this, I know people are saying it's not clear, but because it's hazy, probably it is too small. But I'm telling you that when you go to the browser, even UCSC browser, maybe I can go to UCSC browser today or tomorrow and show you, actually go live and show you how you pick the reading frame when you're doing it by hand. Okay, but you don't have to always do it by hand because I'm going to erase all the mess I made here. Okay, so in this particular gene, the two exons, when you look at it, this is from the reverse frame. This exon is in the sixth frame, this exon in the fourth frame. So the frame you have to know the frame to be able to tell which amino acid is changing. Because remember, when you go to here, the three frames you're seeing, this is not the frame because there is an exon here and the stop codon there. And we had to figure out which reading frame could it be, and we said it was the fifth reading frame, and we said that aspartic acid is changing here, and the codon for aspartic acid was GAC, and GAC becomes a GAA. That's how we found it. The same thing we are doing here, although you guys are saying you are not able to see it, okay, but since I am able to see it, I am saying that this is in the reverse, this is the first frame, in the reverse, this is the third frame. Okay, when the frames are usually set 0, 1 and 2, in that nomenclature, this will be the second frame on the reverse, this will be the 0th frame on the reverse. Okay, so which file, so now you have to have a file which carries information about every exon's reading frame from 20,000 genes, 25,000 genes in human. It has to be a huge file because each 25,000 each gene has, let's say, 3 or 4 exons on the average. So you will have almost 100,000 rows for each exon. That file is called GTF. So with every reference assembly, you have to create a GTF. Again, this is very coordinate dependent. As I told you, if you have a reference assembly that nobody has used before, then you have to create your own GTF file. It's not like you can create a standard GTF file and it will work with every assembly. Every assembly is very coordinate sensitive, and every assembly has to have its own GTF file. So if you're using the older assembly, you cannot use the GTF file from the newer assembly to say, oh my god, my gene is not here, because the coordinates have changed. So make sure that you have the GTF corresponding to the assembly or the reference that you're using for your work. So GTF has to be responsible for that. Okay, so it has to be for a given reference. And you have a start and end. Tens of thousands of exons within the genome will be there in the GTF file. I'll show you how the GTF file looks like in a minute. And then there are tools. I use this tool. There may be other tools out there, but at IBAB we are using, called Snip Effect. Snip Effect uses this GTF file and tells you, creates a database. It creates a database, okay, and uses the database every time you. So SnipF takes a variant call file, a VCF file, okay, VCF file, and you have to use the respective database for your assembly. So that's why sometimes you just got the genome out of the thing. People ask you, is it in the exon, is it in the intron? You can't say it because it takes a lot of downstream work to make this SNP effect database. You have to make a GTF file first. To make GTF file, you have to predict the genes. I told you how you can predict the genes in using Augustus in the second or third lecture. So you predict that, create a GTF file, use that GTF file to and the SnipF utility program to create a database. This has to be done for every assembly. Okay, and then you got to name it properly so you don't by chance use one of the SnipF database for the wrong assembly. Okay, so then when you do that, then you can say, these 300 VCFs, these are the effect. You automated the whole thing. What you did by hand here in this slide, sorry, I went too far. What you did by browsing by eye and everything, now you can completely automate it. For 300 or 1000 or whatever VCF. So every VCF now you can annotate it. When you intersect it, that annotation goes with it. Okay, so the VCF files will now carry annotation after you run SNP effect. Okay, now you can tell, okay, this homozygous mutation, oh my god, you're changing a valine into a glutamic acid and hence has to be deleterious. Okay, one example I'm going to show. How you call something is deleterious or not. Okay, I'm hoping you are with me. We are doing a lot in this lecture. I will take the questions from you tomorrow and maybe we'll do a little bit of quiz to see where you guys are not getting it. Okay, but examples always helps you to understand better. Even if you missed it, pay attention to this example. There is a gene called BRAF, okay, where a valine at the 600th position within that gene is changing it to glutamic acid. And we know that this is deleterious because this mutation is found in 30% of melanoma patients, and this is also a diagnostic for melanoma patients to see whether some drug will work or not. Okay, so when you go to UCSC browser, I don't know whether you guys are familiar with it. We'll do UCSC browser. We have to do it in this course for a short while because I want you to play around with it, or even senior students can take you to the lab and show you. You go and type in to the Google BRAF V600E coordinates on HG19. It will give you the coordinate. Then you take that coordinates and paste it in the UCSC browser. That is exactly what I did, and I'm getting this position. This C goes into a. in this C. It may not be clear here on the screen for you. This is a base C that goes into A, and that, of course, it's calling for a valine. Okay, valine. Okay, when it changes, okay, it becomes a ACC, because, and this is in the opposite direction, CAC, and this becomes a A. Since this is coded in the opposite direction, the codon will be CAA, not CAA this way, CAA this way. So when it becomes a A, then it becomes a AAC. Okay, so this becomes a glutamic acid. So how do you know this is deleterious? Of course, valine is a hydrophobic amino acid, glutamic acid is a hydrophilic acid. So obviously anyone can guess. But the biggest predictor of this is you look at, these are all the species for which UCSC gives you what is happening to each amino acid. Okay, you can see that across the board, even in zebrafish and other things, this is a valine in that gene. Obviously, if it is valine so conserved across all species, it has to be deleterious when you change it to glutamic acid. So this is one of the way you predict the impact of a particular mutation. So a lot of mutations are going to change the amino acid, but not all amino acid proteins are not sensitive to every amino acid in their chain. They are sensitive to few amino acid, which when you change, its function totally changes. And those are absolutely conserved across species. Okay, so that's how you know that the BRAF mutation is deleterious. Okay. I'm going too far. Maybe it will come when it comes to it. I think I'm going to skip this step because maybe I'll start this portion tomorrow. The impact of not using real reference into that. I'm going to skip to sample of a GTF file. Of course, you can see that each of the rows are wrapping around here. Okay, this is wrapping around like this. Yeah. So, and this column is what is keeping the translation frame, the frame. So this particular CDS, this is the coding sequence. CDS is translating in 0th frame. The next CDS of the same gene is translating in frame 1, and the next one. So this is the information SNPF is using. I'm just giving you an example of that. Example of a GTF file. You will probably open a GTF file in your lab and you can see it. So this information is what SNPF is using to get you the right amino acid change for a given variant. Okay. Next bed file, and I'm going to skip. This is a VCF file. I guess you guys have seen it. Again, here is a wrapping each. This is one row, this is one row, this is one row, and it wraps around. Chromosome information here and the coordinate here. Chromosome and coordinate goes together whenever you're merging, intersecting, subtracting. It uses chromosome number has to be exactly same, coordinate has to be exactly same for it to do the arithmetic right. Okay, so here G is the reference allele, T is the alternate allele, then quality, then blah blah blah, all this goes on, which I think people, senior students will run it as to what are all this information that's following each row in a VCF file. So you know the VCF file how it looks like, you know the GTF file how it looks like. GTF file used to create a SNPF database to find the impact of a given VCF. So impact of this G becoming a T, you get it by running the GTF file through SNPF, getting a database and putting this VCF file as an input, then it knows this G to T is happening at the intronic region. Within an intron, so And then it happens within this one, happens within an exon, and then it changes amino acid or synonymous, but are non-synonymous. So, so all this will come from running a SNP program. Okay. As you said, now where we are is now we know how to find homozygous mutation, how to find signatures from homozygous mutation. As this is just a summer camp, I'm just introducing all these concepts that we use in population genetics, and actual methods and everything you will do in hands-on classes. In hands-on classes, you need to be aggressive. If you have a doubt, you got to ask the students because they can be overwhelmed. Okay, if you want to do something, you just be curious and ask them in a nice way. Okay, they'll be able to teach you more than what we had planned to teach here. Okay, so you know where you want to go from this. So that's quality over quality control over generation, and then identification of CRISPR-Cas where there are gaps in 70% of the population where there are gaps. And the last but not the least, which I was afraid I won't have time to talk about, is variant calling is promiscuously used in cancer genomics because using a particular mutation, yes or no, one can give a particular treatment, one can know when a particular treatment will work or not. Okay, so I don't think I'll be able to get to that today. And in cancer, it's not the same thing as population genetics. There's significant difference between how you deal with the variant, how you even identify variants, how do you even sequence for variant. You can't just do 30x coverage of the whole genome and find cancer mutation. You cannot. So the whole thing, whole paradigm changes when you're looking for cancer mutation. Okay, so for example, I'll end it with this, and tomorrow we'll go into somatic variants. How do you find somatic variant in cancer, in the case of cancer? In germline mutation, just to wrap up for today, in germline mutation, since germline mutations are there in half of the reads because you have a diploid genome, and when you fragment it, half of the fragments will come from one arm and half of the fragments will come from the other arm. So if you do say 30x coverage of the genome, 15 reads per base per position will have mutation that's there coming from mother, and 15 will have coming from father. So 30x may be good enough. And you have to do genome wide because you're finding germline mutation. And you're not really looking for any causative mutation or driver mutation and stuff like that. And the good thing about germline mutation, you can just take blood samples because germline mutations are there in every tissue in your body because you inherited that from your parents, embryonic stage, it will be there everywhere. And then after, as you age, you get more mutations in different tissues, but all the tissues will have germline mutation. So that makes it easier. For somatic mutation, they are only there in tumor tissues. You have to have a biopsy tumor tissue to be able to find that. And when you take a tissue, they are often contaminated with normal tissue because depending on where exactly they put the biopsy needle. Okay, from lung cancer, many people just don't, they know the approximate location and they go and pin it. They may sometimes go away from the tumor tissue, not too far away, but a little away. So those samples can have more normal samples than tumor samples. So it's already contaminated on day one. So because of that, you need huge coverage, 1000x to 10,000x coverage. And because you cannot do 10,000x coverage and 1000x coverage whole genome, will really cost you a fortune. No patient is going to pay you that kind of money. So we need a different sequencing strategy, which is called targeted sequencing. This is where we will start tomorrow, and we'll do a little bit. You already know how to call variants and everything, but we'll talk about other strategies we have to use for somatic mutation. Okay. So I don't think I'm going to do any polling today. Um, if you guys want, if you didn't get something, you can type in questions right now. I'm going to wait for 4 minutes or 5 minutes to get all your questions. I won't answer it now, but if you didn't get something, if you got it, then press a one. Say you got it, or if you didn't get it, type in your questions. So I'll collect all the questions. I'll start tomorrow's lecture by answering those things. Okay, I'm going to wait for 4 5 minutes. So for you to frame your question and type it on the chat box. Or if you got it, just say that I got it. I think a lot of people are missing today in the class. Um, Let me see. I don't see Mayuri. Maybe if she's there, she should sign in. Um, yeah, only 8 or 9 of you are there. So I'm hoping you guys are typing in right now. Your question, or you say that yeah, I got it, ma'am. If you got it, say got it, ma'am. Bye. That will be the end of the lecture. Okay, bye now. So I'm going to stop the stream. See you tomorrow.